Micronutrients are an essential nutrient, but needed in smaller quantities. They are measured in parts per million, not percentages like the macronutrients. Soils often have sufficient quantities of micronutrients, but the deficiency is most often due to a lack of availability, not the lack of quantity. pH is a big part of that. The ionic form taken up by plants for copper, Cu2+, iron, ferrous, Fe2+, ferric, Fe3+, manganese, 2+, zinc, Zn2+, or ZnOH2. And for the anions, boron, BOH3-, chlorine, Cl-, then the other cation, molybdenum, MoO4, 2+. The redox potential with the oxygen in the soil, you have the pore spaces. So soils are mixes of big and little pore spaces. And so that means it's also a combination of aerobic and anaerobic conditions. So even in aerobic soils, fine pore spaces can stay anaerobic. So for oxidation, you have ferrous iron going to ferric iron in aerobic conditions. Reduction, you have ferric iron going to ferrous iron in anaerobic conditions. So pH is the biggest driver of micronutrient availability. Deficiencies are usually because of availability. So most often due to pH. So cations increase in acid soils. Anions increase with alkalinity. The greatest availability is between a pH of 6.5 and 6.8. Specific to iron uptake, plants will use either reduction strategy or chelation strategy. In reduction, the hydrogen ion is pumped out of the root. Iron is reduced then at the root's surface. So Fe3+, ferric, is reduced to Fe2+, ferrous, which is then brought across the membrane and into the cytoplasm. In chelation, the ferric is chelated, so it's a ring that wraps around the nutrient so it can be brought across the membrane and into the cytoplasm, where then the chelation is stripped off, and that 3+ is reduced to a 2+. So chelation wraps around the nutrient and facilitates transportation across the membrane. Within the soil or substrate, it protects the nutrient, so pH doesn't affect it. It can stay at 2+, and then the plant strips off the chelation. The 5 factors of soils that impact the micronutrient availability: 1, low organic matter, talking supply here when you have less than 2%. High clay, you're talking again supply, but positively. You have the cation exchange where the nutrients are held, which prevents leaching. 3, you have cool, wet soil conditions, so you have a lack of oxygen, and that feeds in then to the redox potential. So oxidation is the aerobic, reduction is anaerobic. 4, you have competition from microorganisms for the micronutrients. The microbes always win. 5, pH, again the biggest driver. Change to the insoluble form precipitates change to the ionic form, the plant can't take it up. Phytohormones are chemical messengers that have a synthesis and action site. Nutrient supplies both the synthesis and action sites. Auxins are produced in the leaf and act in the roots. They are transported through the phloem. Auxins are produced in the leaf and they act in the roots, transported down through the phloem. Cytokinins are produced in the roots and act in the leaves, transported through the xylem. Cytokinins are produced in the roots and act in the leaves, transported up through the xylem. Low nutrient levels, not deficient, result in low cytokinins and high auxins, which means it has a small shoot system and a big root system. In low nutrient situations, you get low cytokinins, high auxins, which means you get a small shoot system and a big root system. In high nutrient situations, not toxic, you get a decrease in auxins and an increase in cytokinins. Means more shoot, smaller root systems. High nutrient, decrease auxins, increase cytokinins. More shoot, smaller root systems. All nutrients have impacts, but nitrogen is the biggest player in the regulation of phytohormones. Root to shoot ratio. The root dry weight divided by the shoot dry weight gives you information about how the photosynthate is partitioned in the plant. Root dry weight divided by shoot dry weight is the root to shoot ratio. If the root to shoot ratio is less than 1, you get more shoot than root. If it is equal to 1, obviously they are the same. If the root to shoot ratio is greater than 1, that represents more root than shoot. Functional equilibrium is the balance of biomass between the roots and shoots. Functional equilibrium is the balance of biomass between the roots and the shoots. Nutrient levels, nitrogen, phosphorus, magnesium, have the largest impact on altering the root to shoot ratio, a direct influence on photosynthesis. Light, water, and nutrients can all affect the root to shoot ratio. Light, water, and nutrients. Lability. Nutrients are translocated out of the leaves to storage organs and vice versa. The nutrients move around in the plant. Example, sweet potato slits, potatoes. The 3 periods of sync development. 1. Flower induction. Meristem transitions from vegetative to reproductive structures. This is well before you see the flower bud. 2. Pollination. 3. Tuber initiation and growth. These are the 3 critical periods of sync development: flower induction, pollination, tuber initiation, and growth. The source is the photosynthesizing leaves. The sink is the roots, meristems, and fruit. If an organ doesn't transpire, nutrients don't move there well. The meristem and fruit have no stomata and no transpiration, and therefore no water pull. So if the organ does not transpire, the nutrients don't move there well. For example, the meristem and fruit have no stomata and therefore no transpiration, so the water isn't pulled there and the nutrients along with it. Factors that impact yield. You have tech aspects like your agricultural practices, biological aspects like disease, pests, and weeds, and environmental factors like climate, soil fertility, topography, and water quality. So it's the things that meet the crop's needs, the nutrient supply availability, source of nutrition, right things at the right times. What parameters you're driving to the harvest. The quantity and quality of the yield come down to the partitioning of resources, so the photosynthetes, between photosynthesis and respiration. Photosynthesis, the photosynthate partitioning, the rate of photosynthesis, the rate of photosynthesis is influenced by the nutrient supply. Respiration, use of photosynthate to produce ATP. When a plant is growing vegetatively, you can direct photosynthesis with the nutrients. When it begins to flower, it becomes much harder to drive photosynthesis because the plant starts redirecting nutrients to the fruit. So it's a difference, vegetative versus reproductive. 3 responses to increasing fertilizer. 1, maximum quality is achieved before maximum dry weight. Example, snap peas. The quality of yield increases. Decreases as the fertilizer increases. So the maximum quality is achieved before the maximum dry weight. 2, maximum quality is achieved after dry weight, maximum dry weight, like poinsettias. 2, the maximum quality is achieved after maximum dry weight. 3, quantitative and qualitative growth are in sync at the same rate, like lettuce. 3, quantitative and qualitative growth are in sync at the same rate, like lettuce. The 3 responses to increasing fertilizer. The leaf area index. Generally, increasing the leaf area equals an increase in photosynthate, which increases the yield. You have greater light interception, which is how much of the soil is covered by the foliage. Higher than average leaf area index actually reduces the yield and can reduce the quality. It's competing for resources. If you over-fertilize the plant, it pushes out a lot of leaf growth and you get higher than average leaf area index, which can result in a reduction in yield and reduction in quality. Suboptimal nutrient supply situation would see a decrease in leaf growth, a decrease in the leaf area index, decrease in yield. So it would be limited by low photosynthesis, insufficient cell expansion, or both. Suboptimal nutrient supply situation has limited photosynthesis, insufficient cell expansion, or both. Roots are, in fact, a high metabolic cost. They take between 10 to 50% of a plant's daily photosynthetic production. In the dark, roots can't photosynthesize. They make ATP, they respire, metabolize, and they are a necessary expense because they bring in water, nutrients, and anchor the plant. But they are a high cost. They take a lot of the photosynthate produced in the leaves. Let's see, increased leaf nitrogen supply positively affects chlorophyll and rubisco levels. So photosynthetic levels. The nitrogen use efficiency, high photosynthetic capacity increases to a point. The response is plant dependent. Does photosynthesis for all plants respond the same way to increase nitrogen rate? No. Root density is the total length of roots per unit of soil volume. It is a key factor to establishing the soil volume, to estimating the soil volume explored by a root system, and consequently the amount of water and nutrients available to the plant. The root density is the total length of roots per unit of soil volume, and it's a key factor to estimating the soil volume explored by a root system, and consequently the amount of water and nutrients available to the plant. Suboptimal nutrient supply, low photosynthetic rate, insufficient cell expansion, or both. Limiting factors. They are limiting factors. They decrease in leaf growth, decrease the leaf area index, decrease yield. Root interception. As applies to nutrient movement in the soil or substrate and uptake by plants. Ions in solution. The ions in solution and are held on this cation exchange are taken up directly. Root interception is improved by root hairs, which increase how much soil the root system touches. The amount taken up by a plant through interception is affected by available nutrients. How much? How dense is the concentration? What is the root volume as percentage of the soil volume? What is the pore volume as percentage of the soil volume? Roots grow through the pore space. Mass flow. Ions move with water in soil as that water is pulled toward the plant root, toward the plant root through the transpirational stream. It increases as transpiration increases. It is affected by the concentration in the soil or substrate and the volume of water being transpired. Mass flow. The ions move with the water in the soil as that soil is pulled toward the plant roots through the transpirational stream. It increases as transpiration increases, and it is affected by the concentration in the soil and soil and substrate as the volume of water is transpired. Diffusion. Ions move from high concentration to low concentration within the soil or substrate. Top down, side to side, ions move with low solubility. They move slowly and not far from where they're placed. So for example, phosphorus. The amount of nutrient taken up is determined by or affected by the diffusion coefficient. It's related to how soluble the product is, generally estimated by the total uptake of the plant minus the interception and mass flow. Diffusion. The diffusion coefficient is relative to how soluble the product is. Generally, it's estimated as the total uptake by the plant minus the interception and mass flow. Depletion zone. A depletion zone forms around the plant roots due to the uptake of the soil's resources by the roots. The roots compete for what's there. It's an impact of root density. Plants need the appropriate amount or concentration of nutrients. The nutrient uptake and dry matter production changes based on how much nutrient is available and what part of the plant is growing. So, How does the biomass composition of herbaceous plants differ from that of woody plants? Herbaceous plant biomass is 90% water and 10% dry matter. Woody plants, on the other hand, are 50% water and 50% dry matter. Factors that impact biomass production or yield: environmental factors such as light, carbon dioxide, genetics, what is the plant's maximum size and growth potential, water, nutrients, so availability and rate that's present. Annual plants have less stored reserves and they grow continuously. Annual plants need more NPK at a 2:1:2 ratio. Annual plants need more NPK at a 2:1:2 ratio. On the other hand, woody plants require a 2:1:2 or even maybe a 3:1:2 ratio. Woody plants do better with a 4:1:2 or 3:1:2 ratio. When thinking about managing nutrients in different systems, the primary guiding question is, what are our goals? Is the goal aesthetic, the nutritional quality of the seed for the harvest? Is it a container plant? Is aesthetic important and not growing to its full potential in the pot? You have to meet the plant's needs. Different growth rates, growth flushes or cycles, vegetative to reproductive, so the allocation of nutrients change with uptake changes. And is it continuous growth needs versus storing nutrients and reusing? You have to balance the needs of the plant with the expense of the fertilizer. You don't want to waste the nutrient resources, you don't want environmental pollution, and you don't want excessive growth consequences. A lot of our fertilizer sources are not renewable. Toxicity range. Growth falls off with increasing nutrient supply. It doesn't quit growing, just starts growing more slowly. The critical concentration of a nutrient, the nutrient concentration in the tissue needed for maximum growth, maximum genetic potential. Critical deficiency concentration is where the deficiency symptoms begin to be seen. Critical toxicity concentration, the toxicity symptoms begin to be seen. First, deficiency range. In the deficiency range, the growth rate increases with increasing nutrient supply. In the deficiency range, the growth rate increases with increasing nutrient supply. Deficiency range. Growth rate increases with increasing nutrient supply. Adequate range. The growth rate reaches maximum and the maximum is maintained across the range of nutrients. Adequate range. Growth rate reaches the maximum and the maximum is maintained across the range of nutrients. Luxury range. The tissue concentration of nutrients is high enough to sustain plant growth even if the root uptake is inhibited or when growth is very high, so it's storing the nutrients, especially in herbaceous perennials. Luxury range. The tissue concentration of nutrients is high enough to sustain plant growth even if root uptake is inhibited or when growth is very high, there's storing of nutrients, especially in herbaceous perennials. Toxicity range. The growth rate falls off with increasing nutrient supply. It doesn't quit growing, it's just growing more slowly. So, toxicity range. The growth rate falls off with increasing nutrient supply. Critical concentration of a nutrient. The nutrient concentration in the tissue needed for maximum growth, for maximum genetic potential. The critical concentration of a nutrient, the nutrient concentration in the tissue needed for maximum growth, for maximum genetic potential. Critical deficiency concentration, where deficiency symptoms begin to be seen. Critical deficiency concentration, where deficiency symptoms begin to be seen. Critical toxicity concentration, where toxicity symptoms begin to be seen. Critical toxicity concentration, where toxicity symptoms begin to be seen. Between Roman numerals 1 and 2, it's considered deficient. Between 2 and 3, it's low in the nutrient, not critical concentration. 4 is adequate, 5 is high, 6 is toxic.